Hello, everyone, and welcome to today's webinar, High-Throughput Detection of Protein Functional States with Navinci's In Situ Proximity Ligation Assay on the Leica Biosystems BOND RX Fully Automated Research Stainer. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Biosystems. To learn more, visit leicabiosystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize this Q&A box and we will assist you directly. I'd like to now welcome our speaker, Dr. Doroteya Raykova, Application Scientist at Navinci. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Doroteya Raykova, you may now begin your presentation.
Right. Thank you for this introduction and welcome everybody. Welcome to this webinar. As you know now, we are going to talk about ways to detect protein functional states with the in situ proximity ligation method and how the Leica BOND automated stainer can help us do that. My name is Doroteya Raykova. I am application scientist at Navinci, and I have worked for many years in the area of development of molecular tools and in situ proximity ligation-based methods. So yeah, let's start talking about this. So all of you, I assume here, know who Leica are. But I want to say just a couple of words to introduce us at Navinci. We are illuminating function in spatial proteomics. We are a Swedish biotech company located in Uppsala. And what we do is that we develop and provide innovative solutions for studying protein-protein interactions and post-translational modifications. And we have established ourselves as a center of excellence for proximity ligation-based assays. And now that I have mentioned spatial biology, let's take a look at the evolution of spatial biology in recent years. That has really become a big topic, as you know. And I'd say we started by really looking into transcripts with spatial transcriptomics, which offers spatial context on the expression of mRNA transcripts in tissues. It's a really very informative way to look at your tissue. But something that's important to keep in mind is that the expression of a transcript is not directly correlated to the expression of a protein, meaning that just because you have a very highly expressed transcript doesn't necessarily mean that you will also have an equally highly expressed protein encoded by this transcript. So the next stage was then to start looking at proteins in a multiplexed manner. Again, keeping the spatial information, looking at hundreds of proteins at one time. And of course, this was a great step forward because with these many different spatial proteomics platforms that we have available nowadays, we can see the expression of hundreds of proteins at one time. We can see where in the cell they are expressed, and that gives us a lot of information. The downside of this is that just because a protein is expressed doesn't in itself tell us what this protein is doing and what other molecules this protein is engaging with. And in order to exert its functions, a protein must engage with other molecules. And most commonly, this is, a part of cell signaling where proteins undergo different post-translational modifications or interact with other proteins. So even though it's great to look at multiplex protein stains, that doesn't give us information about protein function. And here is where we are going into a kind of a new omics era, spatial interactomics. This is where Navinci excels. So here we have, again, the spatial context, like in the other omics areas that I talked about. We can look at protein expression, but the new additional thing here is that we can assume protein function based on looking at those protein interactions or post-translational modifications. So, for example, we can see when a protein, let's say, for instance, a receptor is binding its ligand. We can see that, for example, now this receptor is getting phosphorylated and downstream signaling is going on. This is a much surer way to say that, okay, this pathway is activated in this cell or in this tissue. So now we can talk about protein function. And many of you might be wondering, okay, it's great to look at a receptor and ligand, for instance. That might be important to understand protein function, but can't I just use my regular immunofluorescence protocol and just simply co-stain for the two interaction partners? And the answer to that is, of course, you can do that. However, if you are like most of us, you don't have a super resolution microscope in your labs, and you have a wide field or a confocal microscope instead. And if that is the case, then you are going to run into resolution problems, because there is something called the diffraction limit that is determined by the wavelength of the fluorophores that you use to stain your proteins. And so, long story short, the best resolution can be achieved with the lower wavelength fluorophores, for example, in the green spectrum. And even with those, the resolution that you can get at best is about 200 nanometers. Which means that any two fluorescent objects that are closer to each other than 200 nanometers will coalesce and appear as one object. And 200 nanometers in molecular terms is a big distance because for comparison, protein complex is more in the realm of 50 nanometers. So you can imagine that something that is 180 nanometers will appear as overlapping. And then you might reach the conclusion that, okay, those two proteins are co-localizing, they are probably interacting with one another. But in fact, that might not be true. So this is all to say that attempting to identify interacting proteins with conventional types of microscopy, such as wide field and confocal, might be misleading, might lead to the detection of false positives. So the question is what to do then to avoid that problem. And the answer is in what we offer, hopefully. I'm talking about the in situ proximity ligation assay, which is a sort of a molecular ruler that comes to the rescue. So with in situ PLA, you can detect, like I already mentioned, protein-protein interactions, post-translational modifications, or why not even single proteins with higher precision. And you get a resolution of about 10 to 14 nanometers without the necessity to have any special imaging instruments in your lab. So you can continue using your wide field microscope or your scanner. You don't need anything special. So I will go over the basic steps of the method. Let's see. So let's say these two orange blobs that you see are your two proteins of interest, protein A, protein B, that you hypothesize are interacting. In the first step, you start by adding primary antibodies. And each antibody is against, one is against protein A, the other one is against protein B. Here I want to just add something in brackets. At Navinci, we aren't an antibody vendor, so we don't sell antibodies. And with the exception of our targeted kits, our flexible products don't include primary antibodies. So you can use whatever antibodies are your favorite ones, the ones that you use for IHC, for IF. Just go ahead, as long as they answer the criteria for in situ PLA compatible antibodies, you can use them. We can discuss what these criteria are a little bit later. But okay, so back to how this works. Use your primary antibodies on the first step, very much like you do in normal immunohistochemistry. As a second step, you have something very similar, again, to IHC. You add secondary antibodies, but we call these Naveni bodies. And the interesting thing about the Naveni bodies is that they are secondaries which are conjugated to oligonucleotide arms, those squiggly lines that you see. So the oligo arms have a specific sequence that gives them this very special structure. They also are different, so each arm is different from the other, but they are complementary. But the complementary sequence or sequences are sort of shielded and can't interact with one another unless the Naveni bodies bind close to one another proximally, which will happen if your primary antibodies bound to epitopes that are within this range of 10 to 40 nanometers. So if you have proximal binding through a series of enzymatic reactions, the protective structure of the oligo arms will be broken. They will be able to interact with their complementary regions, and you will have a formation of a DNA structure looking something like that, forming a DNA circle of sorts. And so that circle, the sequence of that circle, can actually be amplified in the next step in a process called rolling circle amplification that some of you might be familiar with. As a result of this rolling circle amplification process, you end up with a long strand of DNA with a repetitive sequence, the sequence of the circle from step 3. And now that we have this sort of bundle of repetitive DNA, we can add fluorescently labeled detection oligos, which are antisense to that long single-stranded DNA. And in this way, you have hundreds of oligos with hundreds of fluorophores bound to that long DNA. This is how you get amplified signal. Then the next step is simply to visualize your data by using any microscope or scanner. And then an optional step is to also quantify your data. And an example of how the data may look like is this tissue that showed us lots of love. It's a colon, human colon, and the detected interaction here with the in situ proximity ligation method is E-cadherin beta-catenin on both sides. Just as you see on the left side, it's fluorescent detection, and on the right side is HRP-based bright field detection. And this is what the latter is what we will focus mostly today, and how you can use the Leica BOND RX automated stainer to get these really cool images and this important functional information from your tissues. So we have developed a kit which is called Naveni Bright Bond, which is compatible with the BOND RX automated stainer. And so everything is very simplified, so the whole process gets much easier. All you have to do is to dilute your reagents according to the instructions. Basically, insert your slides into the BOND RX automated stainer, and then just call it a day, because you don't really need to intervene anymore from that point on. The bake and dewax step is happening inside the instrument, so you don't need to do that manually. And all the steps, or most of the steps that I showed you from the in situ PLA protocol, are also happening in an automated manner in the machine. So the paraffinization, antigen retrieval, by the way, that is set to ER2, 100 degrees for 40 minutes. But when you're testing a new pair of antibodies, it's a good idea to test if these conditions are optimal. And then the only manual steps that you need to do is, like I said, just to prepare your reagents in the beginning, and then to take out the slides from the machine when the run is over, and dehydrate them, mount them, and image them. And the imaging can be done really on any bright field microscope or scanner, or of course it can be done on Leica's Aperio automated slide scanner. So that also can be automated, but it's a separate step. So with this happy marriage between Navinci and Leica, you can do parallel runs of up to 30 samples. You don't have to do in situ PLA on all of those samples. You can do also immunohistochemistry in parallel. And as I hope to convince you, this is very robust and really reproducible. It's also time-saving, because the total time that takes you to run this protocol on the Leica BOND RX is 9.5 hours. And what we recommend is that you just run it overnight. You can also start in the morning, but it's very easy to run it overnight and just let everything happen while you're resting at home, and then in the morning when you come, you just take your slides out. Whereas when you're doing the manual protocol, it's a longer experience. It's a 2-day thing, perhaps 3-4 hours on day 1, 6-7 hours on day 2. There's a lot of hands-on work, a lot of manual washes that you have to do. It's not the end of the world. It's not so hard to perform, but of course, when all these steps are standardized and automated, you get much more robust and reproducible data between runs and between labs. Also, good news is that we have a standard template of the protocol included in the Leica BOND software. The readout, as the name of the kit suggests, Naveni Bright, is a bright field readout. And what you can use this for is FFPE tissues and also FFPE treated cell samples. So let's talk a little bit about the software. Like I said, the lucky news are that there is a standardized software that is already available, pre-uploaded in the list of standard protocols. So you don't have to program anything yourself. It's very simple. Just choose Naveni Bright from the default list of protocols, and everything will be uploaded. Of course, the Leica BOND RX is an open platform. You can modify steps if you want to do that, but it's really not necessary. Everything is optimized and ready for you. So that's very simple. And let's talk now about validation of the method. So first, let's look at the reproducibility and the variation between labs. So we provided our kit to 3 independent labs, and they were asked to perform this experiment of staining for the PD-1, PD-L1 interaction in human tonsil FFP tissue, which is known to be very positive for this interaction. And you can see here that all 3 labs had very, very comparable results. They got positive stainings in all of their 3 replicates that looked similar not only between runs, between replicates, but also between the labs. And at the bottom, you can see that they also performed a negative control. It was a technical negative control with a B there in the little icon on the side, stands for blocking buffer. So they did not use any primary antibodies. However, they used all other kit components and they ran the protocol in the same way. And of course, this is supposed to be blank, and it is for all 3 labs. So that shows you that the method is very reproducible in between labs and runs. Next, when it comes to validation, you can look at the specificity of staining here. So you might recognize this tissue. It's again the colon. And it was stained for the E-cadherin beta-catenin interaction, which is abundant in colon. So on the top in those zoom-ins, where it says MR, that just indicates what was how the experiment was performed. So MR means that a mouse and a rabbit primary antibodies were used in this experiment. So you have the full staining, and you can see that you have plenty of staining. Everywhere where you can see staining, it means that there is an interaction between E-cadherin and beta-catenin. In the technical negative controls below, the M and the R, one of the primary antibodies was omitted and only the other was used. And if you recall from a couple of slides back when I explained how in situ PLA works, so that circle that is formed and then amplified to give you signal is only formed if the Naveni bodies bind in proximity to one another, which cannot happen if they don't have something to bind to. So in this case, they're missing one of their targets, and this is how you get this technical negative control. And you see the negative controls were really blank, so the specificity is high, the background is low. Next, we also compared how the kit behaves when you run it manually, as it was in the beginning, and how it compares with the automated version that we developed together with Leica. So the automated one is on your left side and the manual on the right. Here we used a tissue microarray, the same tissue microarray for both kits, the manual and the automated one. And this TMA contained both healthy and cancer tissues. The first little picture that you see with that little icon that says BUNC is a biological negative control, which is supposed to be blank, and it is, both in the manual and the automated runs. And you can see that in the rest of the tissues, there is supposed to be staining, and it is very comparable. The localization and the intensity of the staining is very comparable between automated and manual run. We also tested the Naveni Bright BOND RX kit on different tissues and with different combinations of targets. So we have, again, E-cadherin beta-catenin, we have mucin 16 mesothelin in ovarian cancer, we have the CD8 MHC1 interaction indicative of T cell activation. We have PD-1, PD-L1, and ZAP70-LUT in various tissues, both healthy and cancer. So we have tonsil and lymph node as healthy tissues, but we also have many different cancers, pancreatic, skin, ovarian, breast, and so on and so forth. And you can see that with all these conditions, it worked really well. And the staining is strong, clear. Again, let me just reiterate, you can use your own primary antibodies that work for you in other assays. And you can, of course, use your own tissues of interest. That's the idea. Next, let's talk about the workflow, and let's go into like a deep dive of how to successfully set up an in situ PLA assay on the Leica BOND RX automated stainer, but also how to sort of switch our thinking from the traditional looking at single proteins and protein expression into understanding protein function through looking at protein interactions and post-translational modifications. And in the images here on the left-hand side, you can see an immunohistochemistry PD-L1 staining in Hodgkin's lymphoma sample, which is the golden standard, as many of you might know. It's the companion diagnostics for patient stratification for immune checkpoint inhibition therapy. And on the right-hand side, you see a consecutive section of the same Hodgkin lymphoma tissue stained with in situ PLA, not just for the PD-L1 expression, but for the PD-1 interaction, which is an alternative and arguably better way to look at this, because that shows you not just that the proteins are present, but that they are actually interacting, and then you can expect downstream effects from this interaction. So the first step when you're about to start with in situ PLA on the Leica BOND for the first time is to do something that you know very well how to do, namely your pair of antibodies for your proteins A and B that you want to see interacting should be run with a standard immunohistochemistry method on the Leica BOND RX. So this is done to determine the optimal antibody concentration. So titrate each of your primary antibodies by doing this. And also use this to optimize your conditions for dewaxing and antigen retrieval. Because remember that when you're performing in situ PLA, you're using two primary antibodies together that are performing as a pair, which means that both of these antibodies should perform well and in a comparable manner under the dewaxing and antigen retrieval conditions that you use. So it might be that one antibody performs great in both acidic and basic pH when you do the antigen retrieval, but the other one is more sensitive and needs only, let's say, low pH to perform well. So then you should choose that condition that favors both antibodies. Next, with the knowledge that you just acquired from your immunohistochemistry with each individual antibody, use these optimal conditions that you determined in the previous steps and now run the in situ PLA protocol. Like we said, just upload the Naveni Bright protocol from the default list, and then compare your PLA data with your immunohistochemistry data. More often than not, you see similar intensity in staining, potentially lower because you can have higher expression of the individual proteins and lower engagement between them. And of course, you should see the correct localization between the two of them. Like you see here, we have mouse e-cadherin tested on immunohistochemistry. We have beta-catenin, and then we have the combination of the two used for in situ PLA to detect the e-cadherin beta-catenin interaction on the bond. And it looks very comparable. The tissue overview looks the same, and when you zoom in on the lower row, you see that the staining also looks very much the same. Now, what if your individual proteins are both expressed, but you see no interaction signal from your in situ PLA experiment? Well, that can happen. There could be various reasons for that. Here you can see an example. We looked at an FFP prostate cancer tissue, and on the top row we looked at protein A and protein B. You see both of them were expressed, protein B not as highly as protein A. That was done by regular immunohistochemistry on the BOND RX. And then performing the in situ PLA, the interaction of proteins A-B, we got positive staining. But when on the same tissue we looked for protein A and protein C, which also were both expressed, especially protein C very highly expressed, as you can see, we got no interaction. And that is because just because two proteins are expressed doesn't mean that they're interacting. It's one of the points that I made in the beginning. There could be also more trivial explanations for that. The simplest of which is that simply your proteins aren't in proximity. They maybe aren't meant to interact, or they're further away than 40 nanometers. Then, of course, you will get no staining. Then there is also the option of epitopes being hidden. That could be a result of suboptimal dewaxing and antigen retrieval process. And that could also be the result of, for example, your primary antibodies are detecting an epitope in a certain conformation of the protein. Let's say they were developed for Western blots, where the proteins are sort of stretched out. Whereas they have a different conformation when they're in their native state when you perform in situ PLA, and now that epitope doesn't really exist anymore and your antibody can't recognize the protein. It's also possible that the interaction in itself is covering the epitopes that you're trying to detect. And in this case, it's a good idea to just try a different pair of antibodies. There is also the possibility of your primary antibodies not being compatible as a pair. Again, like I mentioned in the previous slides, when you are optimizing the conditions for your antibodies, it could be that they just simply require two different conditions and then they can't perform together. And finally, but frequently a reason for that result is titration effects. So perhaps one or even both of your primaries are at a very low concentration, insufficient to produce results, so you can try to titrate them up. Now, sometimes two wrongs do make a right. What do I mean by that? This is kind of, this slide shows you sort of the opposite situation. So here in this slide, in situ PLA was used for the detection of a single protein, HER2. And why use in situ PLA to detect a single protein? Because it gives you a much more sensitive and specific result. What do I mean by that? Here on the top row, you see tissue from her graded TMA with a grade score of 0. That means that it's not supposed to be positive for HER2 at all. But in the first two images, you see the results of using two different anti-HER2 antibodies with immunohistochemistry. And you see that especially the mouse anti-HER2 antibody gives a lot of background in this HER2 negative tissue. But even the rabbit anti-HER2 also sort of gives a little bit of staining. However, when you combine those two antibodies together and use them in situ PLA to detect HER2, you can get rid of a lot of the off-target effects of the two not so great, not so specific antibodies, because the likelihood of both of them binding the same off-target protein is much lower. So as you can see here on the right side, when you combine the two antibodies and use the Naveni BOND RX kit, you basically get a blank staining, as you should. So even with two not so great antibodies, you actually can get a much more correct and sensitive result. On the row below, you see a different example. This TMA has her score of 3+, so it is positive for HER2 expression. Again, you can see the same mouse and rabbit anti-HER2 antibodies were used in immunohistochemistry. They stained very strongly, especially the mouse, as we can expect. When you combine them for in situ PLA, on the right side, you see this time we actually have staining that shows us there is HER2 present in this tissue. However, not as much as you would think if you just look at the first image where only the mouse anti-HER2 antibody was used. So all of this is basically to say that if you're used to setting up immunohistochemistry assays in your lab, and you're used to using the Leica BOND instrument, then setting up an in situ PLA assay should not be any challenge for you. It's actually pretty straightforward and pretty easy, especially with everything pre-programmed for you. What controls should I use? A frequent question that we get. A very good idea is to use technical negative controls where you omit one or both primary antibodies. This type of control gives you a feeling of how much background you get from your primary antibodies. Another good idea is to just use tissues with a known positive or negative expression of proteins. So biological negative control or a biological positive control, very good idea. You can also use cell lines that have been engineered to have various levels of expression of your proteins of interest. Another option is to exchange one of your primaries with an isotype control. Here, be careful to not use too much of the isotype control, because even the best, most sensitive antibody, if used in excess, is going to start binding unspecifically and giving you background that actually tells you nothing about your assay. And finally, another good control is to use antibodies for an interaction that should not be present in the tissue that you're studying. And just as an example here, we see various different PD-1, PD-L1 interaction stainings on different tissues. So in image A, you can see PD-1, PD-L1 in tonsil. The first image that says MR has both primary antibodies for PD-1 and for PD-L1. And you can see the specific interaction staining in the germinal centers of the tonsils. And the next two images, M and R, are again just those technical negative controls where you omit either one or the other primary antibodies. You see they are negative. In image B, we have 4 different FFP-treated cell lines, each of which has varying levels of PD-L1 expression. So the first one is totally negative for PD-L1. Then we have low, medium, high PD-L1 expression. But neither of those cell lines expresses PD-1. So we are missing one of the interaction partners, and all of these should be negative, which you can see that they are. And finally, in C, we just compare a positive staining, a tissue that is supposed to have the PD-1, PD-L1 interaction, and the adrenal gland, which should be negative for the PD-1, PD-L1 interaction. So that's a biological negative control. So to sum up, when you wonder what controls to use, of course, choose those that are important for your specific question or hypothesis. But as a rule of thumb, always include the technical negative controls, and if you can add biological controls to that, definitely even better. And with that, I hope that I've left you with more knowledge and understanding of how to look at protein function with the in situ PLA method on the Leica BOND RX. And I thank you for your attention. I'd take your questions now, but if you have questions that come to you later, please feel free to contact me on my email address that you can see here, Doroteya Raykova at Navinci.se. And yeah, let's take your questions now. Thank you.
Thank you, Dr. Raykova, for your informative presentation. We will now begin the live Q&A portion of the webinar. We have a lot of questions that have already come in, but if you haven't submitted any questions yet, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question asks, I work with fluorescence. Is it possible to run fluorescence isPLA on the BOND RX?
Yes, great question. Yes, it is technically possible to do that. However, not with this kit that I spoke of in this presentation. So the Naveni Bright BOND RX kit is, as the name suggests, a bright field kit. But otherwise, it's definitely possible to use fluorescence, and actually stay tuned because we are right now working on developing a fluorescent kit that you can use. It's coming soon, and it's going to be really cool.
That sounds amazing. All right, our next question here says, is it only membrane protein interactions that you can detect, or can you also use PLA to see nuclear interactions?
Yeah, there is no limitation when it comes to wearing the cell. The interactions that you're interested in are located. So it works very well for membrane interactions, but it also works for nuclear interactions and for cytoplasmic ones. So there is a permeabilization step in the protocol which allows the antibodies to penetrate the cell. And for nuclear interactions, you might need to permeabilize a little bit longer sometimes, but overall we have optimized this protocol in a way that it kind of works for everything. So yeah, it works fine.
Great, thank you. All right. Our next question asks, do I need to program or make changes to the protocol on the BOND myself?
No, as I mentioned, no programming is needed when you run the Naveni protocols on the BOND. If you want to add an additional staining, then you will have to add container names of these different antibodies in the software and then scan them into the protocol. But yeah, overall, everything is optimized and you don't need to do any programming or changes.
Wonderful, thank you. Okay, next question. Do I need a special imaging system to do this, and what are the requirements?
Nothing special is required. Like I said, for this specific kit, you just need a bright field microscope or a scanner. So you can do this manually, you can do it automated, you can use the Leica scanner. The choice is yours. We aren't bound to any one specific imaging platform, but yeah, and the requirements, just as long as it's a bright field readout, there's nothing special really. You don't need a high resolution microscope. That's one of the benefits of this method that I try to explain, that it gives you this very high resolution down to a single protein or down to a complex of two proteins without the necessity of advanced microscope equipment.
Great, thank you. Okay, our next question asks, how does one choose antibodies for PLA?
Yeah, that's a good question. I intended to go into that in a bit more depth, but I slipped my mind. Yes, so the main thing is that your primary antibodies have to be of two different species. For this specific kit, the Naveni Bright Bond, they have to be mouse and rabbit. Other than that, it's good if they have been validated for use in immunofluorescence or immunohistochemistry. That isn't to say that you can't use other types of antibodies, but these are the most, this is the safest bet that these will work really well for you. But we always recommend that you also test them yourself, just doing an IF or an IHC, as it were, to see how they perform. But that's about it, really. And like I said in the presentation, just run a regular immunohistochemistry on the bond, see how the antibodies perform, titrate to get the optimal concentration, look at the best conditions for both antibodies. That's it. But just have two different species. That's the main.
Okay, good to know. All right, moving along, we have a ton of questions coming in, so please feel free to submit them. If we don't have time for all of them, again, we will get to them via email after the fact. But our next question here says, I'm working with a very low abundance protein and available antibodies are not great. Will this method help me?
Yeah, definitely. I would think so. So yeah, the example I gave with HER2, of course, that's not a low abundance protein, but definitely this is a very good method for that, because like I said, you can use two not so good antibodies. You're saying that yours aren't so specific. You can use two not so great antibodies, usually with PLA that gives you a much more specific result. And because you have this signal amplification step, that actually helps visualize low abundance proteins much better than regular methods. So yeah, should work, I would say.
Great. All right, our next question. How do you know that you have picked up all proximity events and that the assay is not under or overperforming?
Yeah, the in situ PLA method is an antibody-based method, and therefore all the same considerations that you have when you're using, let's say, regular immunohistochemistry are still in power here. So how do you know that you have stained all your proteins when you do an IHC, or that you aren't staining more than what is really there? It's the same thing. You should always think about doing your controls that I spoke about. The technical negative controls give you a good idea of background that you can expect. Some antibodies are better than others, and then of course, if they are very unspecific, they might still give you some background. And it goes without saying that the titer of your antibodies will play a role as well. So if you use very low concentrations of your antibodies, of course, there is a chance that you underperformed, like you put it, so that you don't detect all the interaction events. That's why we recommend that you titrate each antibody individually before you do your isPLA experiment to make sure that you have picked the perfect concentration, which is the best, the balance between the highest signal and the lowest background. So, hope I answered your question.
Yes, I hope so. All right. Moving right along, our next question asks, will there be a webinar for using isPLA with MIF as well?
That's a good question. We haven't planned it at the moment, but certainly if there is interest about that, we can do that. It's very possible to use isPLA with MIF, and we have some posters that show how that can be done and how you can use the PLA method together with other platforms for multiplex protein staining. So you can go to our website, navinci.se, and you can check our resources, and you can see different posters that we showed at the AACR conference. But yeah, we can definitely do a seminar on that. But it's very simple and it's very easy. So, yeah.
All right, great. Our next question here says, is isPLA as good with frozen or FFPE tissues?
So it works with frozen tissues. But for this particular kit that is optimized for the Leica BOND RX stainer, we have validated it for FFP tissues. We haven't validated for frozen tissues. So I cannot say with certainty how that would work or not work. Our manual kits work on frozen and FFP tissues. So.
Great. All right. Our next question here says, can the technique be used to determine homotypic protein interactions, i.e. the same protein interacting with each other?
Well, sky is the limit when it comes to what you can use the technique. I guess it requires some extra thought and ingenuity when it comes to that type of questions. But I can say that I know it has been used, for example, for detection of formation of homodimers. So that can be done. You should be a little bit clever about it, but you can do that. But if you want to discuss exactly how to do that, get in touch with us and we can brainstorm together.
Excellent. Thank you. Our next question here asks, how sensitive is the method?
Yeah, good question. Very sensitive. But again, like I said earlier, it's like any immuno-based assay. It boils down to the quality of your antibodies. And so if you have extremely bad antibodies, I would say isPLA really remedies that to a large extent compared to IHC, to IF, but it still isn't it isn't the answer to all questions of sensitivity. So the better antibodies you have, the better sensitivity you will get. And yeah, I mean, all your preparation steps will affect how good your results are in general. I would say now with this automation protocol on the Leica BOND, that actually helps because everything is so standardized and optimized. But yeah, it's a sensitive method.
Wonderful. Thank you. All right, our next question here is a little lengthy, so bear with me. It says, I have a question regarding the quantitative assessment of the obtained signals. Have you tried performing such an analysis? And for example, during validation, do you also make comparisons between replicates and labs based on quantitative analysis or only qualitative?
Good question. We have definitely done quantitative assessments. I would say that in-house, so far we have mostly focused on such analysis when it comes to fluorescent output, fluorescent staining, because it's a little bit easier. I would say the resolution is slightly better. But it's definitely possible, and we do during validations we do make these comparisons. We are actually thinking about putting out a suggested pipeline for how people can do such quantitative analysis. I also want to point out it's easiest when it comes to cell lines, but we are working on developing something for tissues as well. And it's very valuable for us to know that you guys are interested in that. And yeah, I also want to add, many options are available. You could use free softwares such as Cell Profiler, for instance, or QPath, ImageJ. You can use more sophisticated ones. But let us know what do you use in your labs usually when you do this type of analysis for other purposes, and if you're interested into getting a little bit of a standardized pipeline, and we'll look into that.
All right. We have time for just a couple more questions here, so we'll try to squeeze them in. This next one asks, is it possible to use this kit-based method for DNA-protein interactions?
Yes, I would say that it is possible. Again, so the kit was developed for protein-protein interactions. This would require a little bit of extra work. So yeah, I mean, theoretically it's possible. It's not going to be as simple and straightforward, but I know that it has been done. So my answer would be yes.
Great. And the last question we have time for today, this one asks, do you have other species for secondary antibodies besides mouse and rabbit?
So you mean, well, I presume you're asking if one can use a different combination of species. So the Naveni Bright Bond kit is made to work with mouse and rabbit antibodies. For now, that's it. The manual kits offer some other possibilities as well, not too many, but several others. So you could use, for example, goat rabbit or mouse rabbit. Then, you know, there is also an option to, like, if you, let's say you have some very exotic antibodies, or they don't even need to be that exotic, but not falling into one of these groups, mouse, rabbit, goat. Then we have a kit which is called Naveni Link, which allows you to basically conjugate yourself your primary antibodies to the oligonucleotide arms. So you essentially kind of make your own Naveni bodies yourself. And in this way, you become kind of independent of the antibody species. You could conjugate, for example, 2 rabbit antibodies. And because now they have these 2 different arms, it doesn't matter that they are the same species. But yeah, that is, of course, a modification to the protocol, because then now you will not be, you won't have primary antibody step, Naveni body step, but those 2 steps will become one. And in the manual protocol, that's easy to do. In the bond, I don't really know, but I mean, since that's the RX is a kind of an open platform that you can change things, then I imagine maybe the protocol can be modified to accommodate that. But I haven't tried it myself.
Wonderful. Thank you again, Dr. Doroteya Raykova, for your time today and for introducing us to your important technology. We would also like to thank LabRoots and our sponsor, Leica Biosystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.